Today we will talk regarding the element of visual perception and some human eye construction. I will teach you little bit at the initial slide. Means that more than half of today's lecture is related to biology and the structure of the human eye that we have. Then we will talk regarding the light and electromagnetic spectrum. Color fundamental and image model will be the last part that today we will talk. And then for the next lecture, we will go to more concept of the image processing. As you will see, slowly we will finish the concept regarding the subject that we have, and then uh, we can start our real work slowly by the concept that we will think. Okay, the eye elements in 2D and 3D will be something like this figure, which I can say there's some biologists or some real physician doctor should teach you more. But anyway, um, I also can teach you something related to our subject. Let us see how much I'm successful to do this job. From this figure, the part that I highlighted is more important due to some similarity by image capturing system or image acquisition. More descripting about this part, I already put on lecture note 1. You can read it, but here also I will try to give you more description. This it's important because, as I say, that for the image acquisition and having the view of the first box of image processing, which is acquiring the image, it will help us to analyze it better, and somehow we can say the semi application or semi principle it's happened with the acquisition of data. Okay, and another thing is that the human eye is one of the marvelous things that you can see, because due to the size, due to the application, due to the spectrum of the color, and so many features that your eyes can take it, and analyzation which will be happen based on this vision system. It's I think one of the fantastic part. The left side of you, this is the 3D, and the right side is the 2D part, 2D view of our eye. Let us start from first point, which we call it cornea. Cornea, we call it as the clear lens, which is put it in front of the eyes. This is made by some transparent covering and allows for passage the light into the eye and function as a fixed lens. This, along with passage of the light into our eyes, can also focus the light for our eye. Then the pupil can be the black hole that we have in our eye. I even show you here. You can see in this figure. And when I need more light to enter, when suppose it's dark, the people will do the nice job. Means that it will get larger and allowing more light to enter the eye. And when it's need the less light, when suppose it's bright, it will get smaller and allow less light to enter. Something like some principle like opening and closing the gate for eye to enter and even stop the light for the eye structure. Pupil size is controlled by some fantastic part or some fantastic pump that we can call it iris muscle. And the main function is that the hole where light enters into the eye will be controlled by that. Then maybe just these two parts that I tell you, it will remember you a camera and how you can tune the shutter and how you will capture the figure or picture with the camera. The other parts that we will going to discuss is iris, which is the colored part and circular muscle. And we can say this is the colored part of the eye. The aim of this part is to control the light which will be enter, and amount of that will be controlled by that. Again, you will see some camera concept or shutter concept for the camera. And if please remember, if I say the camera, I speak regarding the previous old camera that we had. Okay, and they also do the same principle and work of the eye, and they try to replicate the eye. Okay. Even this digital camera also have some feature like controlling the entering the light, but think for the old camera that was very difficult, because now there is some auto tune or light adjustment, and the cameras they can work on that. But anyway, in the old time, that was very difficult. Maybe also you, when you were child, you interface that you take some photo and then the amount of light was not proper to that, and then the photo didn't have the good quality. That is something that maybe in our eyes, the iris can control it and also do it better. The sclera is the white part of our eyes, which is the third white skin, which is made of something like tissue, and cover are the eyeball. Means that the all eyeball will be mounted in this part. It supports the eyeball and provide the attachment. The main function which we can say for that, it will support it and provide the attachment of the muscles. Means that you assume it as the chamber which controlled by the muscle, and muscle can make it up and down, and your whole eye can move by that muscle which mounted inside of this chamber. One of the another part which even more important, especially for the vision part, is the lens which is put it behind the pupil, and it's a converging lens, one type of lens that you know, and allow us to see the object near and far. Maybe some of you which have some difficulty to see the object clear, we have the problem for this part, means that the lens part has main problem, which allows us to see the object near and far. Okay, it should tune. Retina is the part which it's the internal membranes of our eyes and contains the light respective cells, rods and cones, and converts the light to the electrical signal. Now something like processors come to picture from our eyes. Is that the amount of light, the pictures, or the light, now it should have some meaning. Okay. The main function of this retina, which we can say it's convert the light waves into the electrical lights. Here I show you the retinal part, and which you can see with the red, the dark red here in the figure, and you can see the position of that. Now, one of the important part is optic nerve, which it's something like wire. It should transmit the electrical impulse from the retina to the brain and creates the blind spot. Brain takes this inverted image and flip it so we can see. Then we should know that the picture that really we will see, it's flip picture Okay, the real one is reverse, and because due to the lenses that we have, and this transmission of electrical signal from retina to brain will happen, and the analyzation will be start. Here is the optic nerve that you can see. Blind spot or retina where optic nerves leads back into the brain will happen, and no rods or cone cells we have. Other eyes compensate for this area will work. It's something like small spot, as you can see in this figure, on the back of the retina, and other eye compensate for this area. It's one of the important part for our eyes, which going to act and help us. Okay, let us see once again this visual perception of our eye. Um, we can see the human eyes at the spur with the average diameter of 20 millimeter. Now, here, after that brief introduction, I will give you more idea regarding the size of the eyes, which it will show you that how much this part from our body is nice, and it will become more clear for you that what we have in our body. It's enclosed by three membranes of cornea, sclera, and choroid and retina. Cornea is the taut and transparent tissue, as we say, which covering the surface of the eye. Sclera is the opaque member, which enclosing the remainder of the eye globe, and choroid is contains the blood vessel to supply the nutrients to the eyes. It's a heavily pigmented, stopping external lights, and it's divided into clarity body and iris. And center opening of the iris that we have, which we know it as the pupil, it's about 2 to 8 millimeter in diameter. And the front of iris is filled with uh, visible pigments, and it's packed with the black pigments. Uh, More, if I wanted to give you the detail, some biological detail, I can say that the lens, which is the fibrous cells, it's 60% to 70% of H2O, 6% of fat, and it's slightly yellowishly pigmented. The retina is rich with the cones and rods, which are light receptors, and the cones 6 to 7 miles in count are permanently located in the center of retina, which we call it as fovea, and they are responsible for photonic, is that bright light vision color. We have some rods that it has the 75 to 150 millions in count and are distributed all over the retina, and they are responsible for the scopic, means that dim lights and vision contrast. And individual cone is connected to individual optical nerves, and hence accounts for the perception of finer details. The group of these rods is connected to group of some optical nerves, and hence account for the ovular perception. The part of the blind spot is in the eye is entirely the deeper of the light per reception rods and cones. Uh, we can say the human eyes is something like the camera, due to the functionality that we have, and the lens form of image on the light sensitive screen, which we can call it retina. Light enters the eye through the thin membranes, which we call it cornea, and cornea forms the transparent bulbs on the front surface of our eyeball. Um, I tell you that eyeball is approximately has the diameter of the 2.3 centimeter, And most of the reflection of the light ray enter the eye occurs at the outer surface of the cornea. And iris, we can call it as the dark macular diaphragm behind the cornea, and it's control the size of the pupil, and pupil regulate and control the amount of light which will be entering the eye. As you can see here in these slides, we focus on the work of eye or principle of our eye. And little bit, if you see, I compare it with the camera or image acquisition system, which we will talk later on that. But if you want to follow the lectures, you will see that for the first part, I just tell you the place of the parts. Second part of a slide, I will give you some biological information. And here, this is some information, some, we can say, engineering information that we need. Biological engineering information regarding the eyes that we have. Some important parts of our eye is the eye lens, which forms an inverted real image of the object on the retina. This is very important and nice things that we have. Means that where we can see in our on eye system that the converted image, how it will be translated for our brain, that again it will flip and then you will see. The eyelash is, as I tell you, it's composed of the fibrous jelly, something like material, and its curvature can be modified to some extent by ciliary muscles. As we said, this lens can be small and big, or can be focused, something like that. The retina is delicate, the membranes having the enormous number of light, and it has some sensitive cells, or we can say that it has some sensitive sensors for that. Then the light sensitive cells get activated upon the illumination and generate the electrical signal, and these signals are sent to the brain via the optic nerves, and brains will intercept this signal and finally process the information. That was what function and principle we have in our eyes. But let us now come closer and see it, suppose, how these lenses will work. As here I show for you, you have two lenses that we call it cornea and lens. The cornea, which is in as the front surface of the eye, does most of the focusing in our eyes, and the lens provide the adjustable and fine tuning of the focus. Then there is two lenses which will be act to have the pictures and help each other to have the picture for us. Muscles in eye can change the shape of the lens, allowing of us focusing on near or far object. Okay, an image is focused onto the retina, existing the rods and cones, and send signal to the brain. Let us now see it from some mathematical point of view. Suppose we have some object like tree, that it has the 15 meter and we are 100 meter away about that object. You will see that our eyes from the lens to the spot point, we have around 70 millimeter in that. We have about 70 millimeter in inside of ours. And the distance between the center of the lens and the retina, which we call it focal length, and varies from the approximately 70 millimeter to about 40 millimeter distance that we have here. If we have the h as the height of the object, h perceived by an observer and having the focal point as the f, which is the um, we can put it as the f, big F, and distance, which we can assume it as the d, then we can have this equation with that h divided by d capital is equal to small h divided by f. Then if I want to know the real height of one object inside my eye, we can have something like this. And this is by the math. Okay. Then you can now calculate, suppose 50 meter divided by 100 meter multiplied by 70 millimeter. Then you will see the size and how the big things can come and sit inside our eyes, and you can have the vision. And my friend have this idea that vision is one thing, and interpretation or elaboration of that is even much more important. It means that the brain is also have one critical part that I'm not going to enter to that part. Otherwise, we should change our course and name it as biology. But anyway, you can read through the notes that I give for you about some parts and functionality of the hair. Okay, then how our lens will be focused means that we have the small depths of fields on our lens, and you can see something close and far with both objects in focus at the time. That is why when we will see two things that they have the near side, one will be focused and another one will come at the background. Some experiment like this you can put. You can hold on your thumb about a foot away from your eyes. Then alternatively, you can focus on the thumb and suppose this figure. Uh, you will see that you cannot see the both the figure means that the screen, computer or mobile screen, and also your thumb sharply at the same time. Then you focus on one of others by changing the bulk of your lens. With some period of time, you can focus on them. Um, then something will come to picture that how we can adjust and adapt the brightness inside of eyes. Uh, it's important because maybe if today you will try to make if today you try to make one camera or any image acquisition system, how you should tune the brightness of the camera. The brightness adoption is some phenomenon which describe the ability of our eye in simultaneously discriminating the distinct intensity level. And the adoption level is the current sensitivity level of the visual system for any given sets of conditions. The simultaneous contrast, we can call it, is the phenomenon which describes that the perceived brightness of retina in an image is not simple function of its intensity, rather it depends on intensities of neighboring region. Some subjective of this brightness is we can call it as some logarithm function of incidence light intensity. And by this plot, you can see that human visual system can perceive about 10 rises 10 different light intensity level. However, at any one time, we can only discriminate between the much smaller number brightness adaptation. Similarly, perceived intensity of a region is related to the light intensities of the region surrounding that. Have this figure in your mind, then we'll talk later. We have some word which we call it match band, and which these words are adjacently spaced. The rectangular strips of constant intensities to demonstrate the phenomena of the simultaneous constraint. Here you will see some example of a simultaneous constraint. All of the inner square have the same intensity, but the other part have the darker and background become lighter, as you will see here. Then even here you will see that from the dark to the gray, we can have the different level of the brightness that is shown in the figure. Some optical illusion we have, which in these eyes fields, the non-existing information or wrongly produced geometrical properties of objects, suppose illumination of the white square, illumination of white circle, illumination of white circle again we have here, but you will see that here these are the dark line or black line, and here you have the gray color line. And elimination of laws of the parallelism and other part from the shape that you can see here, we have this illusion of the eye. Okay, that was some idea that we can take regarding our eyes and some biological principle as well as dimension and data which we need. But really our course is not a biological course that we should focus more on this part. But on the other side, the similarity between the principle of the eye and uh, image acquisition system force us to study and have even the eye structure itself. It's one of the um, fantastic section or part of the body, which if you like you can read more by the net and biological book. What really we need, which is related to our subject, will be start from here, which will be a start from the light and electromagnetic spectrum. Light, we can call it just the particular part of the electromagnetic spectrum that can be sensed by the human eye. The electromagnetic spectrum is called the split of according to the wavelength of different form of energy. Maybe you remember the figure that we said from the gamma rays, X-ray, UV ray, infra, TV, and radio band. Then the light can be split between these bands of energy. What we have, we can call it as the reflect of the light and electromagnetic spectrum. The colors human perceive are determined by nature of the light which will be reflected from an object. Maybe you test this if you want to distinguish between the suppose two blue color, which one color is lighter and another one is darker one. Just by showing the color, you can specify them clearly. Okay. For example, if white light contains all the wavelengths is shown onto the green object or blue object, it's absorb most wavelength and absorb the except green wavelength color, as you will see. Suppose I show here, there is one white light which has the all form of the light range to one object, and here you will see that uh, the observer or eyes can sense the green color by the reflected light to the object. This figure we shown before, but here maybe it's uh, has more meaning. You will see that the radio type of the wavelength, and we are near to this, means that by human, 10 rises 10 or 10 rises 8. Even if we want to convert it into the temperature, it's below that minus 272 of Celsius, or near to 1 Kelvin of the temperature, means that the energy that you can take. And suppose you will see that atomic nuclei, it's how much it has the frequency of the lights. Then these are the electromagnetic spectrum radiation full form, and which it will give us some information about which domain we can see and which totally we cannot see them. Maybe if we were in this state, we could see the virus, or we could be there's some a live MRI system, something like that. But anyway, these are humans. Frequency that it can sense, and electromagnetic radiation part. And we have some concept which we call it monochromatic light, which void of the color, and intensity is only attributes from the black to white. Monochromatic image are referred as the gray scale image, and the light band for this type of the color light, it's from 0.43 to 0.79 micrometer. The quality of this source light of radiance is total amount of energy, and luminance, or LM, which is the amount of energy which perceives from the light source. And one more thing that we need for that, this is the brightness, which is the subjective of descriptor of the light perception that is impossible to measure. And even embodies the achromatic and notion of intensity, and one of the key factor in describing the color sensation. Think for these three parameters that I tell you: radiance, luminance, and brightness. Further, we will work for these words, and we have lots of work to do with these parts. Then, till now, what we learn about the eyes, we learn about light and spectrum analysis. When we learn about some chromatic source of light, let us see what components of image processing system we have. In whole, we can say the image processing, or IP system, is the combination of different elements which they are going to involve the digital image processing for us. And digital image processing of an image by means of some digital computers. This is something that we aim. Digital image processing will uses the different computer algorithm to perform the image processing on digital image. Maybe now you will understand why I give you the previous task for reading about the digital image processing algorithm. Here I give you some brief about these components because whole our course is thinking and aimed for digital image processing systems. But in the lecture note, there is more descripting about this system. In all, we can say that we have these elements: image sensor, image processing hardware. We need one computer to interpret. Then this computer should have the hard copy device, some image display, monitor like that, some mass storage, and it should have some image processing software. Then maybe it can be the camera, maybe it can be your mobile cell phone, maybe it can be your MRI system, maybe it will be some CCT camera, and um, any other image processing system. Let us describe in very brief about this point that what they are, what we can say, consist and which part they have. Suppose the image sensors, which they are sense the intensity, amplitude, coordinates, and other feature of the image and passes the result to the image processing hardware. And it's include the problem domain. The other part is image processing hardware, which is dedicate the hardware that is used to process the instruction obtained from the image sensor, and it pass the result to generate the purpose computer. The other part is the computer, which used to something like general purpose computer that is used by users for our daily life. It can be the software, which is nowadays is much more important part, include all the mechanism and algorithm that is used. Mass storage is the pixel of the image during the process. Hardware, hard copy device, which is once image are processed, then it should be stored in hard copy device, and it can be pen drive or any external RAM device. And image display which is include the monitor or display the screen that display the process image. Then next part is the network, which is the connection all above elements of image processing together. What we learned, that was that in your task, I will tell you what was the aim to describing the I principle and working. Second, we learn something about the wave and light. Now, one of the critical things that we have in image processing is color. Why? Because truly speaking, working with color previous time, that was very difficult in image processing. And even we have some algorithm that we are going to monocolor, gray and black. And then after that, they found that so many things will be missed. Then they start to do the color image processing part, which due to the storage and due to the memory capacity, that was difficult for the algorithm to do it. And you know, the processing of the color itself, previous time, it takes a long time. Now it's become much more better by the advanced algorithm which come to picture. But anyway, first let us say what is chromatic light, which spans the electromagnetic spectrum from the approximately 400 to 700 nano. And as we mentioned before, human color vision is achieved just 6 to 7 million cones in each eyes. Okay, this is the domains that we can see for the colors. A chromatic light that we described before, it's consists of the black and white, and Then the chromatic light colors have their shades. Okay. Um, as I tell you, we have three basic quantities which used to describe the quality of the chromatic lights, which we call it radiance, luminance, and brightness. And we talked regarding these three before. And those are the sensors in eyes responsible for color vision. Approximately, we have 65% of all cones are sensitive to red light, 33% which is sensitive to the green light, and only just 2% is sensitive to the blue. Something maybe come to your mind that why we have the some definition. Maybe you heard before about it. We call it RGB or three main color, which we call it red, green, and blue. You know that all the color in the world are mixture of these three critical color. Maybe when you were in the kindergarten also, they teach you how to mix these three colors. Suppose green and red, if you mix green and blue, green and suppose red and blue, red and green, something like that, mixture of that. And you will take the different color with the help of these three color. Then due to this observation, the characteristics of the human eye, color are will be the variable combination of so-called RGB or red, green, and blue. Then that is why we have three different critical um, um, or not critical, we can call it main color. Okay. The characteristic generally used to distinguish one color from another by the brightness, hue, and saturation. Okay. Then let us see what is these word means. Brightness is the chromatic nation of intensity. Hue is associated with the dominant wavelength in mixture of the light waves, and saturation refers to the relative purity or amount of the white light which mix in the hue. Then review once again. We have RGB, we have BUS, BHS, or uh, brightness, hue, and saturation, which are the more critical characteristics of the RGB color. Hue and saturation taken together and will make one new definition, which we call it chromaticity. Okay, then I even bold these words. I will make this PPT better also, split it. Then you should know these meanings, these concepts well. Then we will work on them. Any algorithm, even truly speaking, that we are working in colors or in image processing, we are interfere with this brightness, hue, saturation, chromaticity, and RGB. Okay, have it in your mind that we will talk more. Then the amount of the red, green, and blue need to form from any particular color. We call it trimestidius values, which are denoted in X, Y, and Z. And if we want to formulate these, we can define as these X divided by XYZ means that the plus of all, y also, z also, like what we will show here. Then a color model which it's come through these previous concepts, or sometimes they will call the color space or color system, is some specification of coordination system and subspace Within that, the system where each color is represented by the single point. In the sense that RGB color model, each color appears in its primary spectrum components of the red, green, and blue. And this model is based on Cartesian coordinate system. What is this coordinate system? It's something like this. Let's okay. You will see here I have the coordination system between the blue, red, and green, which based on this 3D coordination system or cube, I can have the some value of RGB. Suppose if you see the blue, I have 0, 0, 1, means that I have pure blue color. And if you see the red, I have the pure red color. And if you see the here, I have the pure green color. Okay. Then if I want something, some color like yellow, I have some value from the red, some value from green, and some value from blue. Okay. Or if I want the white one, same things. Some cyan or some magenta color. It's also like it. See this point. Okay. Any of them can be the value or having the some value or some mixture of these three colors. Then the color subspace is the cube in which RGB value are at three corners, as I show here. And the three corners are black is at the origin. Means that in black, what I have 0, 0, 0. Means that no blue, no red, no green. It's become at the black. Then if any of these value having any value between 0 to 1 or better things that 255, okay, we have definition for that, then some new color we generate. The gray scale point of is the point which we have the equal value of RGB, means that the value for that gray scale is 111 and extend from the black to the white, means that white is here, then black is here. The gray scale is something somehow here, as you will see here. And in that, we have the equal value for R and G and B. And along the diagonal line, we'll joining these 2 points, which give us the distance or some line between the black and white color. The different colors are points are or inside the cubes and are defined by the vector which extending from the original. R values of RGB are assumed to be in the range of 0 and 1. Okay, that was the cube. Then the matrix of RGB color models well suited for hardware implementation and match nicely with the fact that human eyes is strongly perspective to red, green, blue primary colors. For determining the RGB color model, not well suited for describing the colors in terms that practical for human interpretation. And now we have one model which we call it HSI, which a color perceived by the human eyes is described by its hue, saturation, and intensity. Then HSI, which it's abbreviation of the hue, saturation, and intensity color model, thus decouples the intensity components from the color carrying information of hue and saturation. Then keep it in your mind what is HSI. Let us see this HSI color model, which coordination of that and corresponding color is shown in this figure. The RGB color cubes rotate such that the cube is sending on its black vertex with the white vertex directly above the cyan and blue, green, red, and yellow, and magnitude the vertices forming the hexagon as we will show here. The dot which you will see in this cube is our arbitrary color point, and the range from the red axis gives the hue, and the length of the vectors is saturation. The intensity of all colors in any of these plane is given by the position of the plane on the vertical intensity axis. Have this RGB cube and HSI cube in your mind, and then you will see that we have something like this: blue, cyan, green, yellow, red, and magenta. And then you have some 3D form of them as I shown here for you. Okay, then now you learn the main color and why we have this main color and why even we talk for the red color.